Let's look at the components of the nervous system. The big one that you think of first is your brain. This nervous tissue organ is the control center. The other big part is the spinal cord. This bundle of nerve fibers connects your body to your brain and also has some control of its own. The ones you might be less familiar with are the nerve cells, which are excitable cells, and the ganglia, which are not. A nerve cell is excitable. Anything can set off an action potential. Just like these little lambs, if anything happens, off they go. So the sensory nerve receptors in your fingertips that are just sitting there waiting to detect pressure, they're waiting and waiting and waiting. As soon as your finger sets down on something and they detect pressure, they shoot off that impulse right away up to the spinal cord and the brain. So they're very excitable, and right away they'll send off an action impulse. This is not like your other cells, and not like your ganglia. Your ganglia cells are the nursemaids, the support for all those excitable little lambs that keep running around. All they do is they stay in one spot and they take care of and support the nerve cells. The nerve cells that we're talking about are called neurons. These are nerve cells that transmit information. The characteristics of a neuron is that they are excitable, very responsive to any kind of stimulus. They are conductive. They can conduct bioelectrical signals to other cells. And they can secrete. This is where they release those neurotransmitters that we talked about, like acetylcholine, to communicate with other cells like the muscle cells. These three characteristics, being excitable, conductive, and secretion, are not characteristics that most cells we've looked at so far had. They don't contract like our muscle cells do. They're different from our connective tissue cells, our epithelial cells, our muscle cells. These are the characteristics that define them and make them nerve cells. Your neurons can live and function for a lifetime. They die when the body that supports them dies. In order to study this, they wanted to really find out. They took mice who typically would live around 2 years, and whenever they removed their nerve cells and put those into the bodies of rats that typically lived 5, the nerves kept living past the average lifespan of that mouse, well past it. They apparently were doing fine right up until the rat that it was transplanted into died. So those nerve cells outlived the time span of their host. If you could just keep providing nourishment to your nerve cells, they would keep living even after you died. Nerve cells can live and function for a lifetime, but they don't divide. After fetal development, most neurons lose their ability to undergo mitosis. So if yours become damaged, the body does not replace them. They also have a very high metabolic rate. Your nerve cells alone require abundant oxygen and glucose to be able to function. In one day, they consume about 300 calories. I think that means if you're studying and you're really hungry, maybe that's the way your nervous system is telling you that you need to give it more snacks and glucose. The big 3 of the neuron components are the cell body, or soma. This large area contains the nucleus, and it's the control center of that neuron. The dendrites are short, numerous branches that come off of the cell body that receive information. They bring that information into the cell body of the neuron. A long, single axon is going to conduct that impulse away to the other cells. Here's a different view. The cell body is on the left, with dendrites bringing in sensory information. That information is carried on the axon away from the cell. where it meets up with another neuron, takes that information in, and sends it down its axon. We're just carrying that information along. The cytoplasm of a nerve cell is called the perikaryon. The cell body contains the nucleus and receives input from the dendrites. It also contains Neasel bodies. These are granules of rough ER for protein production. Then we have the axon coming off of the cell body. The definition of an axon is a long process emanating from the cell body. Sometimes it splits into branches called the axon collaterals. Sensory input, which are called action potentials, come in from the dendrites to the cell body. There they collect in a triangular region called the axon hillock. Once enough of them gather in the hillock area, it sets off the action potential, which shoots down the axon. Traveling down the axon, when you get to the end, is called the telodendria. That's where the axon terminates. The synaptic knob is the tip of the telodendria. That's where all of those neurotransmitters that we saw are located. It allows the electrical impulse to be passed on chemically to the next cell. The synaptic knob that we've seen previously was a part of the neuromuscular junction. That was where the nerve cell connected to a muscle cell. But that's not all they do. This example is of a nerve cell connecting with another nerve cell. It will use neurotransmitters to transmit that impulse to the cell in much the same way. There are several zones for a neuron. The dendrite zone, up at the top of the cell body where the dendrites are located, is called the input zone. The axon hillock is called the summation zone. The axon itself is called the conduction zone because it conducts the impulse down the long axon. The telodendria and the synaptic knobs are the end of the axon, and they're called the output zone. Let's review the parts of a neuron that we've learned so far. Can you find the dendrites, the cell body, the nucleus, the axon hillock, the axon itself, and the Neasel bodies? There's a lot going on in one picture. Now, what we haven't talked about are all of these little dark dots surrounding the neuron. What's up with that? What are all those spots? Those are the ganglia nursemaids we mentioned. Axons are very special because they have to travel so far through the body, sometimes up to a meter if it's one that comes from your spinal cord all the way to your fingertips. Because they're so long, it's not possible for the cell body itself to provide nourishment to that long axon. Instead, we have to use those ganglia. They're there to conduct impulses, but without protection, they won't carry that impulse very far. Your body does a process called myelination. It wraps your axons with a substance called myelin. Just like in our own electrical systems, we coat our electrical wires with plastic. The reason we do that is to protect them, to make sure nothing else metallic comes in contact with them and diffuses the electrical current. Instead, we wrap them in plastic to protect them. Your body does something very similar with those long axons. Your body does a process called myelination. It wraps your axons with a substance called myelin. This myelin sheath that is produced is from ganglion cells called Schwann cells that wrap around the axon and form an insulating barrier. It's crucial for the nerve impulse to be able to travel that long distance. On number 1, we're able to see this Schwann cell coming up to the axon and starting to encircle it. It will encircle it many, many, many times, forming a very protective barrier around that axon. Here's an actual microscopic view of an axon. In blue on the left is the axon. The middle picture shows the myelin sheath surrounding the axon, and on the right we see the entire Schwann cell that has completely encircled the axon, insulating it as it conducts its electrical impulse. In the peripheral nervous system, many Schwann cells have to work together to wrap a single axon because each one can only wrap 1 millimeter of the axon. If we're talking about that meter-long axon, you can see it would take many Schwann cells, maybe over 100, to wrap one axon. In the central nervous system, it's easier. They're not having to travel those long distances, so we see these oligodendrocytes that are able to wrap part of their membrane around multiple axons at once. Have you ever seen a picture of the brain and noticed that there was a color difference? That is actually from the myelin. Your white matter in your brain is myelinated neurons. The high lipid content of myelin gives it a glossy white appearance. Areas of the brain where, because the distance is so short, the neurons are not typically myelinated is a darker color. It's called gray matter. By looking at this picture, you can tell that the majority of the brain is highly myelinated. That is to allow all of your nerve impulses to be conducted through your brain. If something happens to that myelin and it starts to break down, those Schwann cells or the oligodendrocytes can't function properly, then you could lose the ability to think, to control your body, to move. All of those impulses from the sensory and the motor can't travel properly through the brain if that myelin starts to break down. Multiple sclerosis is the progressive demyelination of neurons in the central nervous system. It's an autoimmune disorder where the body starts to attack the oligodendrocytes. As it destroys the oligodendrocytes, the myelin around the axons get broken down, and when that happens, the impulses are no longer able to travel. You may have noticed that in all of these pictures, the myelin is not continuous. There's gaps. These gaps in the insulation of the myelin are very important for stimulus transmission. The little tiny gaps in between the myelin are called the nodes of Ranvier, and the stimulus will jump from node to node.